Hi students, in this video you are going to learn about the cell cycle and mitosis. Alright, I hope you still remember some of the materials that we have discussed in grade 10 because that's going to be very useful to help you to understand this topic. Okay, so these are the life cycle of human and cell. Why I talk about the life cycle of human? Because it helps you to understand as an analogy for the life cycle of a cell. So let's look at to the life cycle of a human. Everyone starts his life or her life as a baby, and then this baby has to grow, and as they reach puberty, it means that they are ready to create family. Okay, and then when they create family, it means that the woman has to get pregnant, all right? And then, well, the new baby born will again repeat the cycle, all right? Now, the same thing also happens in the life cycle of a cell. Look at this. For example, these are two cells that were just born, and then each of them will grow, getting bigger and bigger, and then one day they are ready to split into two. Or in another word, we say that they are reproduced. All right, see, these are the two cells that are ready to be split and create the new generation. So that's a life cycle. Born, grow, reproduce. The same thing also happens to human. Born, grow, reproduce, give birth, grow, reproduce, and so on. Okay? Now let's combine the two life cycle into the life cycle of a cell. Again, not really the same, but this just to help you to understand by using the analogy of human life cycle. All right, so we will see the life cycle of a cell. The life cycle of a cell consists of interphase, okay, and then mitotic phase, okay, or this is the place where reproduction happens. All right, so now interphase basically are the growth stage. It is the longest part of life in the life of a cell. All right, this is the longest part. As you can see, this part takes about 90% of the life cycle of a cell. Now let's take a look at the phases, smaller subphases in the interphase. All right, G1. This is G1. Well, I can use the analogy of from baby grow into adolescent. So this is the growth stage as the cell is just born and still small. So like a baby, it's still small and it needs to grow. This phase is very active metabolically. Some cells, however, stop here if they are no longer meant to be more in number. So why it's very active metabolically? Because they need to grow like a baby, like when you are a teenager, you must have a very active metabolic processes. You eat a lot, you grow a lot. The same thing happens to the G1 of a cell. And some cells stop. For example, the nerve cells. Until a certain age, the nerve cell will not divide anymore. That's why old people start to degenerate in terms of their capability to think, or the brain capacity is start to slow down because why? No more new cells are produced. All right, it means that the nerve cells in that old people are actually stopped in G1. They are no longer proceed to the next step. The next subphase of interphase is called the S phase. S phase is actually the DNA synthesis. As you can see from here, DNA synthesis. Now, I hope you still remember our genetic topic in grade 10 that from one cell to another, as they do the cell division, the DNA as genetic material has to be duplicated and shared. So this actually happened in interphase subphase S. All genetic materials has been duplicated in this stage. The cell now is capable to create two cells with identical genetic material or genetic information. Why? Because all the DNA has been duplicated. All right, so it's kind of like this. Yeah, like the adult or adolescent people, they are actually mature and ready to be, get married and have a new family, continue with their life cycle. And then the last phase, subphase, the last subphase of interphase is G2. it is the time to prepare for the cell division, preparation for the cell division. For example, the cell will make sure that the protein required for mitosis are available and so on. Well, the analogy is like this. Of course, when you reach your puberty, even though you have the capacity to create new offspring, you will not get married that soon, right? You have to prepare, like in this picture. Well, maybe you want to have a car before you got married. Maybe you want to have a house. Well, perhaps that's a good idea. Otherwise, where you are going to live. Anyway, and then some of, savings in the bank so that you are ready to get married and have family. Now, after the cell are ready in G2 to do cell division, then they will enter to the mitotic phase. Now, as you can see from here, the mitotic phase itself consists of two smaller phase, which is the mitosis and cytokinesis. By definition, mitosis is division of nucleus, right? So this is a cell. The division of nucleus is mitosis, while cytokinesis is the division of cytoplasm. We will see the two processes in detail in the next slides. Well, seeing is believing. This is the real video of human embryo. See, okay, from 1 become 2, 3. Now, see, you can see that those are the cells, the embryo cells that are growing. Yeah, they are doing mitosis. This is real human embryo from a fertility clinic. So in diagram, we can tell you that this is what happened in mitosis and cytokinesis. So this is starting with G2 and then in entering the prophase, okay, prometaphase, which means actually this is part of the prophase. This is just early, and then this is the later one, and then metaphase, anaphase, and telophase. Okay, as you can see, the product of mitosis is two daughter cells. All right, and you can see also from here, the first step is the division of nucleus. The nucleus has to be gone, and then you can see the genetic material or chromosome will be divided. All right, I hope you still remember your material in grade 10, right? And each sister chromatids will be separated into different poles. Don't worry, this is just at a glance, especially for new students. We will talk about the process in detail step by step after this. Oh, by the way, before you learn mitosis, I hope I can refresh your memory about the genetic materials, because in mitosis, remember, that's the division of nucleus, which means the division of genetic materials. So let's see, this is a nucleus. Nucleus contains genetic materials in form of DNA. So it looks like this. Looks like noodle, right? A bowl of noodle. Now, if we zoom in the chromatin, this is called chromatin, by the way, the genetic materials that looks like noodle. This chromatin, if you still remember, this is eukaryotes for sure. They wrapped up protein histone. So they are not naked DNA. Compared to prokaryote, if you still remember, there is no protein associated with DNA. However, in this second figure, you can see that the DNA wrap around protein histone, right? Like this. Okay, so this, what I say, DNA coils around protein. Well, while prokaryotes do not have the same condition, all right? Now, the loosely coiled DNA is called chromatin. So like this. So basically, those things, which is DNA wrapped around the protein, when it is loosely packed, it's called chromatin. That looks like noodle. However, when the cell do mitosis, of course, it is so inefficient to have DNA and protein that very, very loosely. It will cause tangled and so on, right? Now, to solve the problem, they create a super coiled chromatin, or we call it condensed chromosome, right? Or simply say chromosome. Okay, the process that happens from chromatin to chromosome, that's what we call super coiling. Okay, very, very condensed. Now, this densely packed DNA and protein is no longer called chromatin, but it is now chromosome. So, chromatin, whenever it is experience what we call supercoiling, it will become chromosome. Okay. Now, the next question is why chromosomes are in X shape during mitosis? Like what you can see from here. This is a picture from electron microscope. All right. So, chromosomes supposed to be like this. But if you remember, during the S phase, all genetic materials have been duplicated. So, as a result, when those chromatin that has been duplicated, condense or supercoil, they will create a structure called sister chromatids, which are basically identical copy of chromosome that stand side by side and bind together. All right. Now, who bind them together? This is a structure called centromere. Another new terminology for you. All right. Now, this centromere later on during the mitosis, they are going to break apart, and then the chromosome, the sister chromatids, okay, will be back into a single chromosome. All right. So thus during mitosis, the identical copies of each chromosome can be separated, one for each daughter cell. Okay, the identical copies of chromosome called sister chromatids. Okay, like twins. And sister chromatids are joined together, and the spot is called centromere. This one. So as you can see, at the beginning of mitosis, all right, all chromatin will be condensed and they will become chromosome. All right, this is the prophase, the beginning of mitosis. All chromatin will become chromosome. Okay. Please be patient. Before we enter mitosis, again, one thing. Okay, you have learned about the chromatin become chromosome, and at the beginning it consists of identical copy of chromosome called sister chromatid that attach together in the centromere. One more thing that you have to know is about the centriole. All right, centriole are basically cell organelle that looks like this, and from centriole there comes fibers, or we call it microtubules. Well, basically they are fibers that you can see this arrow that later on will help the sister chromatids to be separated, because the function of these spindle microtubules, by the way, the microtubules is called spindle microtubules. Please memorize those terminology, right? This spindle microtubules will attach to the centromere of the sister chromatids. So later on you can guess that this spindle microtubules will break apart the centromere so that each of the sister chromatids can be pulled apart into the opposite pole. Okay, so now let's talk about the cell division, mitosis. So mitosis consists of prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. So this is prophase. So the chromatin will become chromosomes that consist of sister chromatids. We have learned about it. And this centriole, all right? This centriole will start to produce the spindle microtubules. And at the same time, the nucleus membranes start to disappear. Okay, start to disappear. Now look at this. The centriole will move to the opposite pole, and the microtubules will getting longer. Right? Can you see? The microtubules is getting longer. Okay, let's continue. Now we are entering metaphase. Meta, middle. So you can guess what will happen? Yeah. The chromosome in form of sister chromatids will align at the center. All right, align at the center in the middle. Okay, while the spindle microtubules keep on growing. Let's see what happened next. Okay, see, the spindle microtubule now reach the centromere of each sister chromatid. They attach. Okay, let's see what happened next. We are entering now the next phase. Okay, this is the attachment, which is the anaphase. Now, what happened in anaphase? Yeah, the centromere will break apart because of the spindle microtubules that pull them apart. See, now each sister chromatids, or well, basically now they are already become the single chromosome, are pulled apart into the opposite pole. Okay, this is called anaphase. And the last one is telophase. The telophase. Okay, let's see what happened in telophase now. Okay, the chromosomes start to become chromatin again, and the spindle fiber from the centriole start to disappear, and it also actually telophase cannot be separated from cytokinesis or separation of cytoplasm. As you can see here, it starts to happen. See? Okay. Yeah. I hope you understand about the mitosis. Please allow me to summarize the mitosis. The first step is prophase. All right. In prophase, the chromatin condense and become chromosomes, supercoil, and then chromosome appear as sister chromatids with centromere. Okay, attach them together, and then nucleus membrane start to disappear. As you can see, these are the remaining part of the nucleus membrane, and spindle microtubules grow out from centrioles, all right, this fiber, and then centrioles and together with, of course, growing microtubules that come out from them, move to the opposite poles, one to the north, one to the south. And in metaphase, after prophase, here comes metaphase. All right, in metaphase, chromosomes in form of sister chromatids are aligned the equator or metaphase plate in the middle of the cell here. Right, and then spindle microtubules from both poles are attached to each centromere from the opposite side or on the opposite side. So look at this from here. Yeah, see this attached to the centromere, and then from the other pole, this is the spindle microtubules also attached to the centromere of these sister chromatids. And here comes the anaphase. During anaphase, centromeres have been divided and sister chromatids are separated. As you can see from here, now the centromere has broken apart, split, so that each of sister chromatids, which is now called as a chromosome, move to the opposite poles. And the last stage of mitosis, the telophase, the spindle microtubules break apart. As you can see now, they are short. All right, and all chromosomes have reached the poles, opposite poles. All right, one on the top, one at the bottom, or north and south. And nucleus membrane start to reappear surround them. As you can see here, nucleus membrane start to reappear, and cytokinesis takes place at the same time. So to be honest, like telophase cannot really be separated from cytokinesis because cytokinesis kind of happens at the same time like telophase. As you can see now, the plasma membrane start to get into the center and separating the cytoplasm of these two identical cells. Okay, be patient, please. One more, all right? The syllabus asks you to learn also the difference in cytokinesis between animal and plant cells, all right? Why? Because remember, plant cells have cell walls, so there is no way for the plant cell to be separated in the same style or the same way as the animal cell. So let's take a look at the animal cells first. So this is what happened during telophase. Cytokinesis happened at the same time. So the plasma membrane will go to the center, all right? And there is a kind of like microfiber, microfilaments that will separate them. And this is create the shape looks like cleavage, and is called cleavage furrow, right? And later on, this daughter cell will be separated. However, in the plant cells, as you can see, if the plasma membrane separated but not the cell wall, of course, it's not going to be created into two cells. So what happened is, in the middle of a cell, there are fascicles. Remember fascicle, like in the endocytosis and exocytosis? It will create a cell plate. What is cell plate? So cell plate, the fascicle looks like this. They contain the material for the cell wall, and this will later on join Okay, for example, I have lots of fascicle with the material for cell wall. All right, later on they will grow and combine. Now they are called the cell plate. You can see from here the fascicle is no longer exist, but instead they have become a long strand or long partition of the cell wall. And later on, the end of each side of this cell plate will combine with the old cell wall, okay, which means that now they will separate the cell into two. All right, I hope you understand this. Okay, I would like to thank to all the resources that I have used for the in making the video, and students, I hope you understand this concept. Thank you and see you later.